Cursor, right?
I think that we're going to go ahead and get started, everyone. Good afternoon, and on behalf of Nick Langhals and Ned Talley, my name is Matt Raymond. I'm a program analyst at NINDS. And I want to thank everyone for logging in for our second webinar in a series featuring BRAIN Initiative-funded PIs for developing technologies for recording and modulating neural activity. We have two great presentations this afternoon, and I want to thank our two presenters, Chris Zhu from Cornell and Mark Schnitzer from Stanford. A few housekeeping items before we get started. Everyone who's logged in will be muted during the presentation. We'll have 10 minutes for Q&A following the presentation where the speakers will be able to ask each other questions. And we've also asked Andreas Tolias to act as a discussant as well. And with that, I'm going to turn it over to our first presenter, Chris Zhu, who will be talking to us about multiphoton deep imaging of the mouse brain.
Thank you, Matt. Welcome to the webcast. Good afternoon and good morning on the West Coast. So I'll talk about imaging of mouse brain structure and function, very deep. First, acknowledgement. This work is mostly done by two of my graduate students, Ken Yu and Moran, and also two of my postdocs, Dimitri and David. We have lots of collaborators in the process who really helped us on various aspects, and some companies I work with. Most of the work is done by funding from NIH, and recently we also have a fair amount of luck with DARPA and IARPA. So a brief introduction of myself. I got my PhD at Cornell. You can see me in 1993, a lot younger, a lot more hair. And I spent 5 years in Bell Labs, mostly doing fiber optics. Came back to Cornell, does both biomedical imaging and fiber optics. And for brain imaging, we really started pretty late, around 2008. We started to dabble into how to image the brain, particularly how to go deep. And you can see some of the images in my career in Bell Labs and also in Cornell back a few years ago. Brief motivation. Why do you want to go deep into the brain? And this is a brain talk, so it's very much a no-brainer. You can see this shows here a mouse brain, about a cubic centimeter. And this little yellow box is how much we can do with high spatial resolution imaging. You can see on the left-hand side, about a few hundred microns across and maybe 700 microns deep. So the goal of this proposal is really to see, can we go much deeper into the mouse brain? In particular, we use the hippocampus, the SP layer of the hippocampus, as a benchmark to see how deep we can go in an intact mouse brain. So our goal is to achieve high temporal resolution, hopefully about 10 hertz, and high spatial resolution, submicron, and going deep around 1 millimeter or more in an intact mouse brain. And this is the aim of our Brain Initiative grant. So what's limiting high spatial resolution imaging? Scattering is really the major problem. So here you can see I put a lens paper in front of my cell phone camera. I took a photograph, and you can see you don't know where this is. And you look at it, this is my office, right? You can see the windows, the tables, and everything. You can see photons on the left-hand side. You can see photons do get through and reach the camera. But on the other hand, with lots of scattering, you don't have any high spatial resolution information. Now look at the mouse brain. If you think about this, the mouse brain underneath a wide field microscope with craniotomy. So we remove the bone, put a piece of glass window on the top. You really have to think about looking through a piece of tofu or a piece of cheese. Imagine to see a single cell underneath those things. It's going to be difficult. And the tool we use is called multiphoton microscopy. And this tool actually invented about 2.5 decades ago by Winfried Denk and Walt Webb at Cornell, right here inside the lab I'm occupying right now. So what is the challenge? What are the challenges for imaging deep into a mouse brain? Tissue scattering is really the biggest problem. And we can separate the issue into several different aspects. First problem is that you can see we take a lens, we focus into, say, a piece of brain tissue. What scattering does is that it significantly reduces the amount of photons reaching the focal volume, showing up over here in the black dot. Essentially, every photon, as it propagates towards the focal volume, every time it encounters a scattering event, that photon will be removed. So that photon can no longer reach the focal volume. And this process goes exponentially. So as you go deep into the tissue, the amount of signal you can generate goes down exponentially as a function of imaging depth. That's a huge problem. It's an exponential problem. And experimentally, you can see this. This is a mouse brain vasculature. We stain the blood vessels with a dye. Blood circulates, all the vasculature in the brain are stained. You can see we can go pretty deep from the surface of the brain all the way to perhaps 1.6 millimeter. But on the other side, if you quantitatively look at this, you look at log of the signal as a function of imaging depth. You can see the signal goes down exactly exponentially. And if you go deep enough, as you can see, from the surface all the way to about 1.5, 1.6 millimeters down, we're talking about signal decay of 5 orders of magnitude. So you can see this is a huge problem caused by tissue scattering. And second problem is that you can see the brain tissue are not optical material. They have heterogeneity, they aberrate, they degrade the optical waveform. So in that case, we call optical aberration. And what optical aberration does is that it decreases the signal generation and also makes the resolution worse. And lastly, even if I got excitation happen deep in the brain, in the focal volume, the fluorescence photon still come back out and reach all the way to a detector somewhere outside. And by going through lots of tissue, the amount of signal collection is reduced. If you quantify this, you can see that the first issue is a huge problem, 5 orders of magnitude. The second issue is about 1 order of magnitude. And the last issue perhaps a little bit less than 1 order of magnitude. So the imaging deep is really very difficult. And the strategy we've been using is actually very simple. We go to long wavelength to penetrate deeper, and then we use 3-photon excitation to better confine the focal volume. So just one slide for a little more detail about why this works well. So it's all about the ability to form a sharp focus deep in the brain. So let's look at tissue attenuation. So we go long wavelength. In those long wavelength region, for example, above 1200 nanometer, water absorption really dominates for the brain tissue. On the other side, if you follow the red curve you can see over here, as you go to longer and longer wavelength, the scattering length actually increases. So less scattering by going to long wavelength. That's the red curve. It is the same reason why you see sky is blue, sunset is red. If you combine those two effects together, absorption in the blue, scattering in the red, you end up with a green curve over here, which shows you the effective tissue attenuation length as a function of excitation wavelength. The longer, the better. You can see that clearly 1300 nanometer window and 1700 nanometer window are quite optimal for deep tissue penetration. And these 3 stars are experimental data obtained by various groups, including ourselves. And you can see the simple model works quite well for explaining tissue attenuation in the brain. The second strategy we use is using 3-photon, that is high-order nonlinearity for excitation. So why that's better? It's all about confinement. So let's look at, for example, we focus deep in the sample, say about 1.2 millimeter, 1.3 millimeter over here. You can see that for 2 photons away from the focal point right here, the 2-photon signal goes down as 1 over z squared, where the z is the distance away from the focal plane. If you go to high order, because nonlinearity is higher, away from the focal point, you can see that the decay is much faster. Instead of quadratic, inverse quadratic, I have a 4th power decay as a function of distance away from the focal point. Now, when you put scattering into the equation, this long-range suppression of background fluorescence is really important, because now you have this exponential decay, showing this graph over here. You have exponential decay, right? And you have the geometric focus. Now you can see, away from the focal plane, we have lots of scattering. The amount of background generation in 2 photon actually is quite appreciable. Now if you go to 3 photon, you can see the red curve over here, right? The long-range suppression of out-of-focus background really pays off by going to high-order nonlinear excitation. So again, we use long wavelength to reduce the impact of tissue scattering, and we use high-order nonlinearity to suppress the out-of-focus background. So it turns out these two techniques, the ideas, long wavelength and 3 photon, are very much goes hand in hand. And the reason is that if you want to do 2 photon excitation as those 2 optimal wavelengths for penetration, we don't have many dyes to look at. In fact, we have very few fluorophores to look at those 2 long wavelength windows for 2 photon pumping. If you go to 3 photon pumping, you take the wavelength divided by 3, you can see you have blue-green fluorophores. GFP or GCAP naturally works well for 3 photon at 1300 nanometer window. And if you look at orange and the red fluorophores, over here we look at fluorescent proteins. You can see, look at the wavelength. You multiply the 1 photon wavelength by 3, you very naturally land in the 1700 nanometer spectral window. So in many ways we can say that if you want to use long wavelength and still be practical, you have to use 3 photon excitation. And if you want to do 3 photon excitation using conventional fluorophores, you kind of naturally land in those 2 long wavelength windows. So they're very much synergistic ideas. So I give you a couple of slides showing you the long wavelength and 3 photon advantage. So this one shows just vasculature imaging, 2 photon excitation at 920 nanometer with fluorescing dextran, and 3 photon excitation at 1320 nanometer of the same fluorescing dextran. So this is the same mouse, we image the same place at the same time. At this point, it's about 840 microns in from the surface of the brain. You can see the difference in image contrast. In the 3 photon excitation, blood vessels are very bright, and outside of blood vessel is very dark. And look at 2 photon excitation, you can still see the blood vessel. But on the other hand, you can also see that away from the blood vessels, you still have lots of background signals. If you quantify this, you can see, if you go deep enough, long wavelength and 3 photon wins in absolute signal generation because the longer wavelength attenuates much slower. You can see both 2 photon excitation in the purple and 3 photon excitation in the blue attenuates exponentially as a function of imaging depth. But you can see the attenuation is much slower because the wavelength is much longer. So at the surface, of course, 2 photon is much brighter. But if we go deep enough, 3 photon signal actually can be stronger than 2 photon signal because the attenuation of long wavelength is less. If you look at the contrast, it's even more striking. We just take a line profile across those 2 blood vessels. Again, same mouse, same place, same time. For 2 photon is in the blue, as you can see. It's the blood vessel in the middle. Away from the blood vessel, you can see still lots of background signal. In fact, your SBR, signal to background ratio, is only about a factor of 2 here. For 3 photons, you can see this orange curve, even go 840 microns in, SBR is still about a factor of 100. In fact, we see almost no decay from the surface all the way down to more than a millimeter. Essentially, the SBR is the same. So 3 photon long wavelength also wins in contrast at depth. The same story goes for neurons. So in this case, we look at in vivo imaging of deep, just above the wire matter, layer 6 neurons labeled with GCAM6. Again, the same mouse, same place, imaging place, and same time. So on the left panel showing you 3 photon imaging at 1300, you can see lots of details of the neurons. You can also see the neurons sort of get squeezed within the white matter over here. And you can see the shadow of the blood vessel completely black right here, the 0 point. If you do 2 photon, again, the same place, same mouse, you can see the contrast is much worse. Now this is a virus-injected mouse. We don't even have that much labeling above it. But on the other hand, you can already see the amount of background generation away from those neurons. So you can see that this dark blood vessel shadow in 2-photon is almost invisible because of the smearing effect of the background. So again, we can see significant contrast improvement or background reduction by using 3-photon excitation at the long wavelength. Of course, we want to look for brain activity. So in this slide, showing you a 2-photon excitation, a 3-photon excitation of the same layer 4 neurons stained by GCAM6S. So you can see the image over here. It's the same place, same time. You can see the neurons by 3-photon excitation in the bottom, 2-photon excitation on the top. We do time division multiplexing here. And you can see the activity trace obtained by 2-photon on the top and 3-photon in the bottom. We essentially get exactly the same activity trace for layer 4 neuron. That means regardless of the excitation modality we use, the activity we report from the same neuron here are the same. That's sort of obvious to see. Now I'll show you some more activity imaging for deep layers of neurons, and this is imaging the spontaneous activity of layer 4 neurons. We call deep layer 4 neurons because these neurons are right above the white matter. As you can see on the still image over here on the right-hand side, the blue are the third harmonic generation of the white matter. Densely packed axons actually give you very nice third harmonic generation. So we know exactly these neurons are right above the white matter. And the imaging conditions are in the lower right. You can see frame rate, pixel values, field of view. It's a 16-week-old mouse. We use total of 36 milliwatts to obtain the movie I show you next on the left-hand side. It's a 10-minute movie sped up by a factor of 60. So you can see the neurons flickering. Again, these are the deep layer 6 neurons using GCAM6 excited by 3-photon at 1300 nanometers. And success rate is almost 100%. So almost every mouse we looked at, we see the layer 6 very easily. And you can also quantify that by segment the neurons out, and the data shown right underneath are raw data without any processing. So these are raw data in photons per neurons per frame, and recorded about 8.5 frames per second. I think these are actually a very nice way to show the data, because this way we can compare across different platforms, different 2-photon or 3-photon excitation, different probes. By doing this way, we know exactly how much noise, how much fundamental noise we have in those activity recordings. So showing you here are photon numbers per frame at 8.5 frames per second. That means photons here, about 1700 photons per second from each neuron. So this is the bright neuron, this is the dimmest neuron, this is all the neuron in between. Of course, most of you presented like this. You do low-pass filtering, about 0.5 seconds low-pass filtering. You can see neuronal activity of maybe 40, 50 neurons in this field of view over here. This is again for layer 6 imaging. Now we want to image all the way down to more than a millimeter to see the hippocampal layer. In this case, we want to reach the SP layer in the hippocampus. And it turns out imaging the hippocampus is significantly harder than imaging, for example, layer 6 over here. Because to reach the hippocampus, you really have to image through the white matter over here. Okay? And the white matter is much more scattering than the gray matter. In fact, you can see our quantitative measurement. Again, you can see the fluorescence as a function of depth. We're imaging through the neocortex, the gray matter, imaging through the white matter, reaching the hippocampus. You can see the different decay slope between the gray matter and the white matter. And if you quantify this, you can see that the white matter is about 2.5 times more scattering than the gray matter. That means even though you only have a couple hundred microns thick of the white matter, they actually worth hundreds of microns of gray matter because they're just so much more scattering than the gray matter. So I show you data for the hippocampus imaging at the 1 millimeter. This is the SP layer in the mouse. Again, GCAM6S labeled 3-photon excitation at 1300 nanometer. This is a 17-week-old mouse, a 200 by 200 micron field of view. So you can see very nice structure of those GCAM6 labeled neurons. In fact, I want to point out, if you look carefully, you can see the nuclear exclusion of these neurons, right? You can see donut shape of lots and lots of neurons. See, the fact that we can see nuclear exclusion all the way down the hippocampus without adaptive optics means that our resolution is still very good because we can discern the nucleus from the cytosol. And on the right-hand side, I show you the movie. It's a 15-minute movie recording and sped up by a factor of 20. So you can watch mouse thinking really quite deeply. Right? You can see the flashes of these neurons. Within this field of view, I think we counted something like 140 neurons, and roughly about a third of them showing you very nice activities. And 2/3 are not very active. So you can see the activity movies on the right-hand side. And all this is done in an intact mouse brain, so the superficial layers are not perturbed. We can also quantify this activity in the HASP layer by drawing, again, fluorescence as a function of time. Again, showing here the raw data in photons per neuron per frame. And you can see on the top, this is the dimmest neuron. In the bottom is one of the brightest neurons, so the photon count's about a factor of 3 to 4 difference. In any case, we can record very nice activities with very good signal-to-noise ratio from these neurons. And on the right panel, showing you the low-pass filter with about 0.6 second low-pass filtering to show you the activity traces recorded by those GCAP6. We can also use the same technology to imaging through unthinned and intact mouse skull bone. So showing you here is a three-dimensional reconstruction of the skull and the brain tissue together. So you can see, for example, this panel over here. The green stuff are through harmonic generation of the osteocytes in the skull. So that's the intact mouse head bone. Underneath of this is Texas red stained vasculature. So in this case, we leave the bone unthinned and intact. On the right-hand side, you will see the movie. Again, the function of imaging depth, showing you in the Z. So osteocytes in the bone, in the green. And then you move into the brain tissue. You see the vasculature. So even with the skull intact, you can still see that we can get very good spatial resolution, clearly resolve each individual capillaries. You can see the penetrating vessels or horizontal vessels. We can go almost as deep as about 700 microns with the head bone intact in this case. We can also do the same thing by imaging red fluorescent protein labeled neurons through intact skull. So the three-dimensional reconstruction showing on the left panel, you can see that the red dots are the neurons labeled by RFP, and the green, this stuff over here, again on the osteocyte in the bone, and the green stuff in the tissue mostly are blood vessels and also myelinated axons. So on the right-hand side, you can see different panels of neurons in the red, and through harmonic generation in the green. Again, through harmonic generation by the osteocytes, and the red dots are RFP neurons. And of course, we can image it repeatedly over days, and we can find the same neurons at the same place. And I think this could be very useful by combining other techniques such as a clear skull cap preparation. By our ability to imaging through the skull, you can imagine, will allow us to image almost anywhere along the mouse head. So you don't have the restriction of a cranial window. And we can also combine this technique with low resolution optical imaging or MRI type of imaging. The more obvious question is that perhaps we can use adaptive optics to improve, in particular, through skull imaging. And I'll give you a couple of examples we have done. So adaptive optics, if you're not familiar with, actually is a very simple concept developed first in astronomy, is that if you imagine focusing of light, right? So over here you have a lens, a plane wave coming in, come to a perfect focus. But if a plane wave coming in, after it's going through a sort of heterogeneous tissue, a perfect wavefront will get distorted. And that's the origin of optical aberration, which reduces your signal, also reduces your resolution. Both are bad. So what you do is that instead of having a plane wave coming in, you pre-engineer your wavefront. So what you do is try to cancel out the impact of wavefront distortion by the tissue. So you pre-engineer wavefront, you cancel out this distortion, and then end up with a perfect focus. And by combining this with deep imaging and 3-photon, we expect to have pretty large impact. So I'll give you a couple of examples to show a through focus theory of RFP-labeled neurons in a brain bone mouse through the skull. So you can see on the left panel, no adaptive optics. On the right panel, with adaptive optics. You can see the neurons popping out. On the left panel, it's very hard to see if there's any neurons in this particular depth. And we can quantify this in the next slide, is that you can draw lateral profiles across certain areas in your image. Okay, you can draw this one on the left side is no adaptive optics. On the right-hand side is after adaptive optics correction. And the lower panel is showing you the quantitative analysis of the intensity at the functional position. You can see the blue without adaptive optics, the green with adaptive optics. Obviously, we pick up in a significant amount of contrast also in absolute signal strength by about a factor of 5, maybe 6 or so, depending on which particular side you look at. So adaptive optics can definitely help, particularly in through skull imaging or you want to image really deep. So all this work cannot be done without a robust and optimized excitation source. That's what we need. I'll give you some example here. This is the laser we started with about a year ago, maybe 18 months ago. Okay, that's a big laser made by Coherent called Sapphire Amplified Pump Optical Parametric Amplifier. And little mouse is over here. You can see the little mouse, the size of a knob over here. This is the kind of data we can do in the hippocampus about a year ago in this type of laser. That's the best data we can get. We can still see neuronal activity. These fluctuations are real. It's very noisy because the photon numbers are small. 6 months ago, with the help of Newport Spectral Physics, we got a much better laser system. We also significantly improved our surgical skills to go from being horrible to being, I think, okay-ish. But this is the new data we can get in the hippocampus I just showed you before. Much better data, mouse hippocampus, right? And even better is that I think there's another 2 to 3 times improvement we can expect by re-engineer this laser a little bit differently. So we can do even better than what we have today. And if you want to do this, unfortunately, you need to invest in this laser system. And the good thing is that there are commercially available sources already announced. For example, Spectral Physics announced this about 6 months ago. Coherent just announced 3 months ago, and maybe in another couple of months, they will have this on the market. And both lasers are very good. They're robust. They provide high pulse energy. They're wavelength tunable. Actually, they cover both the 1.3 and the 1.7 micron spectral windows. So you buy one laser, they cover both windows. They're reasonably compact, just 2 boxes, and reasonably user-friendly. I think we can still improve them by going to a little bit higher repetition rate. And of course, they're still pretty expensive. They can reduce the cost. That'll be very helpful. Okay, just to summarize, should we abandon 2-photon imaging? The answer absolutely is no. 2-photon is great if you don't want to go that deep, right? So what's the opportunity for 3-photon imaging at the long wavelength windows of 1.3 and 1.7 microns? I see several opportunities. One is you want to imaging really deep in scattering brain tissue like the mouse brain. I showed you mouse hippocampus. You can certainly move to other deep brain regions. Hippocampus, we use this as an example to show the capability of this technique. You move to bigger animals like the rat or the monkey, you certainly want to go deeper. And for mouse brain GCamp, I think if you go deep enough, 3-photon also wins in terms of absolute signal strength because the decay is less at long wavelength. Even if you don't want to go very deep, if you just want to go layer 5 or maybe top of layer 6 or something like this, if you want to have densely labeled brains, you're going to do volume imaging of the entire cortical column with each neuron labeled, for example. I think 3-photon will also significantly help you because of the suppression of out-of-focus background. And lastly, there's also niche applications. For example, you can penetrate through highly scattering layers. I showed you an example of unthinned intact mouse skull. We have also some success imaging the motor neurons in mouse spinal cord. We can also imaging through thick dura, such as the monkey dura or rat dura, things like this. So with that, I'll end my presentation. Thank you very much for your attention.
All right, thanks a lot, Chris. And so right now we're going to open up the discussion with our two discussants, Andreas and Mark, if you guys want to take the floor.
Okay, thank you, Matthew. Thank you, Chris, for your presentation. I have a couple questions. Maybe Andreas and I will alternate.
Sure.
Chris, my first question concerns indicator development. So as the 3-photon imaging modality is now taking shape, what would you recommend to those of us in the neuroscience community that may be focused on developing activity indicators, and what would you like to see from this portion of the community?
Yeah, so ideally, it would be great if we can make those nonlinear cross-sections much bigger than what we face today. Now that, I think, is a very difficult task to do. But that's really going to revolutionize everything, right? If you can make the 3-photon cross-section just 10 times bigger, right, I can probably reach much deeper into the hippocampus. So that's really one thing to think about.
Right, and then how, do you have specific recommendations as to a good approach to doing that?
Yeah, so that's actually, I don't. There's a lot of work done on 2-photon at this point, right? You know, 2-photon people have been trying to develop large cross-section lines for the last 20, 25 years. And it turns out to be very difficult. I think other than what you like you're doing, you know, screening different proteins for cross-section and all those, I'm not sure there's actually a sort of a theoretical approach to design something from scratch, for example. That's probably very difficult, I think. In terms of wavelengths, I think with the two windows we have at 1.3 and 1.7, I think we can cover most of the moving already very high. The wavelength probably will be okay already.
Great. Just to follow up on that, Chris, so with the development of the red-shifted calcium indicators, for example, how do you see, at least for the cortex, for dense imaging and imaging, let's say, the whole column, what do you think are the limitations of the 2-photon going deep with the red-shifted? What are the advantages and disadvantages of going to 3-photon there?
Absolutely. I think, I compared mostly the green 2-photon at 920 and the 1300 for 3-photon. So if you go to the red dyes, the R-camp are really good. You go to 1.06 micron excitation. I think your performance will be better in the G-camp at 920 nanometer. The only issue is that we actually tried those R-camp at this point. They're not, we haven't, okay, that's the way to put it, we haven't got success in R-camp imaging deep. Not sure why. So it seems like just the red dye is just not as good as the green one at this point.
But that may not be a fundamental issue, right? So,
that's true. It may not be a fundamental issue. Absolutely.
So one, you know, I think Andreas is getting at a good issue here, which is that in principle, one could imagine a complementary and alternative strategy in which you try to develop increasingly red-shifted activity indicators and push more out into the infrared. Now, this, of course, might be complementary with 3-photon excitation, but that would be another possible means of imaging deeper in tissue.
Absolutely. I think in particular, if we can do a 2-photon dye, 2-photon indicator excited by 1300 nanometer, for example, that would be great. I think that may give you a shot even doing 2-photon hippocampus. There may be a chance for doing that. But on the other hand, you still always have this issue about the contrast issues, right? But if you go to hippocampus, we roughly go about 5 scattering lengths. That's roughly the depth we're talking about, even at 1300 nanometer. And if you look at 2-photon excitation, if you have densely labeled samples, that's almost reaching the absolute limit of 2-photon. That makes sense?
Yeah, so basically there is a final limitation, but yeah, I mean, what Mark is saying that...
For example, for layer 6, it might be very logical to do 2-photon at 1300. It's probably perfect for it.
Even for dense imaging, though, even if you have...
Layer 6, we have to see that layer 6 you're talking about is only, at 1300, is only about 3 to 3.5 scattering length. You might do okay. You still have an issue, but it'll be okay. My guess is okay. If you go to hippocampus, you're really pushing all the way to the edge of the 2-photon fundamental limit, right?
Yeah.
Thanks, Andres and Mark. With that, we're going to move on to our next presentation. We're running a little bit behind schedule now, but we're going to plan on sticking around for a little bit after 3 to make sure we get some questions in for our next presentation as well.
Okay.
All right. So I'm Mark Schnitzer from Stanford University. I'm also affiliated with the Howard Hughes Medical Institute. And before coming to Stanford, I actually shared an office for a certain time with Chris at Bell Labs, where we both entered the field of optical brain imaging. So it's a real pleasure to present today with Chris over the web. I'm going to be talking about our work on the development of fluorescent protein indicators of neuronal voltage dynamics, transmembrane voltage dynamics, both the development of new indicators and applications of these indicators. And we are supported in our efforts by NIH BRAIN Initiative Award, a UO1. And my colleague at Stanford, Michael Lin, is also a co-PI. We co-direct this particular project. And we have a subcontractor, Inscopics, Inc., which has commercialized the technology that many of you might have seen before, and fodder from my prior work. You can see here in this first slide a microscope that we're using today for calcium imaging. This is a 2-gram digital fluorescence microscope shown here on the tip of a finger. The mouse that you're seeing is wearing this microscope as it freely explores the enclosure, and meanwhile we're visualizing by 1-photon fluorescence calcium imaging the activity patterns of about 700 hippocampal neurons. And one of our main aspirations in the work that I'll talk about today is that in the future we would like to be able to attain this kind of data at this scope and scale, but with voltage indicators, not just calcium indicators. And that's one of our main future goals for this work. Now, I should also disclose to the audience that I'm a scientific co-founder and a scientific consultant for Inscopics, Inc., and you need to know about that that relationship. Now, from this movie that I'm showing here, there are about, as I said, 700 hippocampal neurons, and we'd like to begin asking the question as to whether we can accomplish voltage imaging across similarly large populations of neurons. So, might we be on the brink of attaining optical voltage imaging in freely behaving animals? And this is, to attain this goal, it's going to require development of new optical voltage indicators, a main focus of our work under this NIH BRAIN Initiative grant. As I'll discuss, it's also going to require some progress in optical hardware, and this is a main reason for the involvement of Inscopics. The present miniature microscope that you see here on the left is not even capable of acquiring image frames at the kilohertz rates that you would need to track single-axis potentials, and to do so would require novel optical hardware. So to attain this goal, there's going to have to be progress in multiple different domains of engineering, protein engineering and also optical hardware engineering. So here's an outline of what I'll be discussing today. To start, I'm going to review the motivation that neuroscientists have for creating genetically encoded voltage indicators, fluorescent indicators that I'll be discussing. I'm then going to spend a little bit of time reviewing concepts from signal detection theory. These concepts will provide useful tools that are going to allow us to benchmark and compare the performance of different optical indicators. And this is an important pursuit because we need to have means of evaluating indicators in a standardized way across different optical microscopes and different experimental preparations. We need to have meaningful ways of comparing indicators across different situations. In addition to the capabilities for benchmarking indicators, signal detection theory is going to provide some useful guides toward the design of new voltage indicators. Third, I'm going to discuss some recent progress in the development of optical voltage indicators, and in particular, I'm going to focus on a class of indicators that my lab has been pursuing, the Fred-Opson voltage indicators, and I'll explain how these work. And then in the last portion of the talk, I will discuss some state-of-the-art usages of optical voltage imaging and some examples that we have been pursuing in our laboratory here at Stanford, including the first instances of detection of single action potential, single spikes in the brains of awake mice. So let's review briefly why neuroscientists have long yearned for optical voltage indicators. So the development of optical voltage indicators began actually in the late 1970s and has continued since then. Initially, indicators came in the form of organic dye molecules. There's been recent excitement in the field from indicators of a different type, the protein indicators, protein fluorescent indicators that can be genetically encoded. And so I'm going to be specifically making comparisons to this subclass of fluorescent voltage indicators, the genetically encoded ones. And the reason that neuroscientists have pinned a lot of hopes on this particular means of recording activity in the brain is that there's the possibility of combining many of the virtues of in vivo electrophysiological recordings with the virtues that we have attained already in the optical field with in vivo fluorescence calcium imaging experiments. Now, both of these other modalities, electrophysiological recordings and calcium imaging, have some important advantages and some important limitations. Extracellular electrode recordings are very, very useful, but are largely limited to cells that have dipole moments of sufficient magnitude. They have excellent capabilities for detecting spikes, excellent capabilities in the temporal domain. But they have some notable weaknesses. The extracellular electrodes do not provide adequate reports of subthreshold neuronal membrane voltage dynamics. There are important challenges in how neuroscientists can target individual neuron types by virtue of their genetic densities or by virtue of their patterns of anatomical connectivity. There are key challenges in maintaining recordings over the long term with extracellular electrodes and sampling dendritic activity, and in dense sampling. We're typically limited by the density of electrode arrays, for example, as they enter the brain. Now, when we consider the advantages and limitations of optical calcium imaging, we have a largely complementary set of strengths and limitations here. Calcium imaging has worked very well in excitatory neurons and allowing neuroscientists to detect the occurrences of action potentials. Calcium imaging has not been as informative for inhibitory neurons, which often fire rapid bursts of spikes, and the limited kinetics of the calcium indicators have confined our ability to follow the individual event trains in many classes of inhibitory neurons. Calcium indicators have a large dynamic range in their signaling capacities, which has provided high fidelity spike detection, but on the other hand, they are limited in their abilities to enable accurate estimation of spike times. So they are much more limited in the temporal domain than the extracellular electrode. They're also limited in their ability to report various subthreshold facets of somatic membrane voltage dynamics, but they do offer the possibility of targeting individual cells by their genetic density or by their pattern of anatomical connections. Long-term imaging experiments have been demonstrated dendritic activity can be monitored with appropriate labeling conditions. And as you saw just a moment ago in some of Chris's movies, one can densely sample tissues, for example, in structures such as neurocortical columns. Now, a main reason there's so much hope pinned on the emergence of new voltage indicators is that it's conceivable that voltage indicators might be able to provide the best of both worlds, providing capabilities in all these different dimensions. And I should say that we're not there yet. Not all of these categories and advantages have been realized yet, but the field has been making rapid progress in recent years, and so there's substantial motivation to continue to have voltage indicators that would provide high temporal resolution, the ability to target cells by particular types, to maintain these recordings over days and weeks, to sample densely, and so forth and so on. Now, one of the main issues that I'm going to be discussing today concerns detection And I want to make a very broad distinction at the beginning here regarding detection versus resolution. This is going to underpin some of the calculations we're going to be doing using signal detection theory. Now, I want to note that in other aspects of optical microscopy, the audience may be familiar with the distinction between detection and resolution, particularly in the context of the super resolution microscopes that you may have seen. In the forms of microscopy based on the stochastic localization of single fluorescent emitters, such as POM and STORM microscopies, you may know that we can detect the presence of individual emitters to a localization accuracy that is far below the traditional resolution limit. And so this distinction between detection and resolution that you may know in the spatial domain also operates in the temporal domain. And so what you're going to see is that in many cases we have the ability to detect neuronal activity as the signals from a given cell rise above the fluorescence background, even if we cannot resolve cells at rest in the images. And this is a key point because today we routinely can have photodetectors capable of single photon sensitivity. So we can detect very small increases in photon flux, provided that the background photon flux that we receive is sufficiently small. And what we'll see is that these background photonic emissions are in fact the chief impediment to detection of neural activity, not our capabilities for resolving individual neurons. I'm going to illustrate this here in the domain of calcium imaging. This is a mouse that's wearing the miniature microscope you saw earlier. It's running back and forth in this linear track. Look at the neuron that's been circled here in the movie, and you'll see that this neuron is not evident when the cell is not firing, but we can see the cell's activity pattern by detecting the increase in photons as the mouse runs through this hippocampal cell's place field. So signal detection theory provides us with a means of benchmarking different indicators, as I was saying before. It's common in the literature to see a variety of different metrics used to describe optical indicators of neural activity. A rudimentary way of describing the performance of an indicator is to look at its dynamic range, the relative change in fluorescence that the indicator undergoes as the neuron transitions from a baseline resting state to an activated state. However, looking at this relative change in fluorescence, often called delta F over F by itself, is an inadequate measure of evaluating indicator performance, first of all, because it does not take into account the fluctuations in the background photon flux. And so in any real situation, we're going to have a finite number of photons emitted from the indicator while the cell is at rest. There's going to be a standard deviation in these fluctuations, and I need to be able to take into account how far above these background fluctuations does the signal rise in the presence of neural activation. This is sometimes called the signal-to-noise ratio in the literature. However, this too is a suboptimal means of characterizing indicator performance. As you'll see, a better means is to make use of the quantity that's often called D prime, the signal detection fidelity that comes out of signal detection theory. And it's superior because it takes into account these different factors, the signaling dynamic range, the fluctuations in the photonic background, and it further takes into account all the photons that we get in the characteristic waveform, not just the peak amplitude change. And it's this entire photon flux during the waveform that we receive when a cell is activated that is going to allow us to make discriminations between a case when a neuron did not fire and a case when it did fire an action potential. So you'll see that the signal detection theory is going to allow us to evaluate how changes in indicator properties can affect our ability to detect spikes and also to time them accurately. We can use it, as I'll allude to briefly, to evaluate new modes of microscopy and actually to benchmark our ability to extract spikes from activity time traces and to provide measures of statistical confidence in the extracted traces of neural activity. So the basic idea here from signal detection theory is that if we have an observation of a given set of photons, how likely is that set of observations to have come from a cell that just fired a spike from a cell that did not? Now, when we see these observations of photons, we don't know the underlying ground truth, but if we run this experiment again and again, we can form the probability distributions that we will see assemblies of photons that are detected that are characteristic of an activated cell versus an unactivated cell. So the difference between the log likelihood ratios in the case when there actually was a spike and when there was not a spike tells us the difficulty of the discrimination problem that we face. And the difficulty of this problem is characterized by the metric d prime, which is a dimensionless measure that tells us the distance between the centroids of these two distributions in units of their common standard deviation. And this is a formalism that goes decades back in signal detection theory. And we can calculate it exactly for our optical cases under the assumption that we are in the shot noise limit, meaning that photon fluctuations are the dominant noise source. And here in this slide, you can see the comparisons between the signal detection theory in the smooth curves and some simulations that we performed of stochastic photon collection. The theory well describes the stochasticity that we see in our simulations and allows us to characterize how difficult the discrimination problem is. Now, one of the things you can see from this graph, or this pair of graphs, is that slight changes in d prime can yield exponential improvements in the error rate. So here on the left, we have a case and where these two probability distributions are separated by only, their centroids are separated by only one standard deviation. And here on the right, we have a case where it's 3. The difficult cases, the difficult ensembles of photons, are those that could plausibly come from either scenario. Maybe the neuron spiked, maybe it didn't. And so the overlap area represents the area of difficulty. And you can see that as we pull these two Gaussian distributions apart, the overlap is becoming exponentially suppressed. And so d prime, which is related typically by polynomial relationships to optical parameters, can be improved modestly through protein engineering, and this can lead to vast improvements in signal detection fidelity. So in any practical experiment, we have to draw a real threshold. What are we going to call a spike? What are we going to say is not a spike? And you can actually vary this threshold. But there's a characteristic of our performance that is inescapable, and this is typically quantified by the receiver operating curve. If we plot our probability of catching true positive, real spikes, versus the false positive rate, there's a fundamental trade-off that is plotted here on the left. And the area under these curves is called the area under the receiver operating curve, the ROC area, and it tells us, given a choice, a binary choice between an instance of a spike and no spike, what are the odds that we can pick the instance that was a spike correctly? And this area under the ROC curve will rise as the d prime metric improves. And again, we can see that small improvements in d prime can lead to meaningful gains in error rates as the d prime value raises. Now, it's important to understand that these choices that we have to make occur at every time frame. So in a voltage imaging experiment, we would typically acquire data at a kilohertz rate if we wish to capture individual action potentials. So this means that we are asking, did we see a spike, did we not see a spike, 1,000 times a second with the kilohertz frame acquisition rates. And so therefore you can see that to avoid having the experiment corrupted with many, many false positives, we need to have excellent d prime values to be able to capture a meaningful depiction of the actual spikes while keeping the false positive rate low. All right. So this is one of the reasons in the voltage imaging field that we have had to seek much higher d prime values than in the calcium imaging field. In the calcium imaging application, one can typically use much coarser time bins. You saw 10 hertz in Chris's presentation just now. And so we asked this question, did we see a spike, did we not see a spike? Many fewer times per second. And so false positives are less of a problem with calcium imaging. Voltage imaging is a more demanding problem. Now, I'm not going to go through all the theory here, but there's some basic ways that we can improve voltage indicators. And this is an approximate relation that can be calculated in the shot noise limited case, again, where quantum fluctuations in photon counts are the limiting factor. And what we see here is that D prime improves linearly with increases in the signaling dynamic range, whereas if we hold the signaling dynamic range constant and we simply increase the brightness of the indicator, we only get a square root improvement in our signal detection fidelity. Moreover, if we're able to keep the background photon flux low, then we can actually attain very high signal detection fidelity, even in the case where we don't have many photons. And so this graph shows you that we can get a very high area under the ROC curve, even in the case that even in the case we have few photons, provided the background labeling is low enough. And so that's going to be an important factor that is going to inform our fluorescence labeling strategies and our indicator design. Now, interestingly, both scanning and non-scanning microscopy modalities can often attain similar D prime values at modest depths into tissue. Now, when new indicators come out, such as those that we're trying to develop, we can use measured kinetic constants and signaling dynamic ranges to estimate D prime values. And so here's a paper in the domain of calcium imaging from Sam Wong's lab at Princeton, publishing different constants here for calcium indicators. We can use these values to evaluate the signal detection fidelity of these different indicators. We can also use this framework to compare different microscopy modalities regarding the performance of indicators in different situations. And these kind of ideas also allow us to tell us whether the algorithms we're using for extraction of spikes from our optical traces are reaching the physical limit set by photon fluctuations and photon statistics. And they will also provide us measures of statistical confidence in the extracted spikes that we get out from the optical traces. So this entire framework informs our approach to optical voltage indicator development. It also provides some indications of how well we might expect to time the action potentials, not just detect them. So it's one thing to detect an action potential, it's another thing to time it accurately. And the limitations we have in the temporal domain with calcium imaging will illustrate this. Now, there are different classes of genetically encoded voltage indicators that have been explored. A number of indicators involve proteins with 4 transmembrane domains. And to give you some examples, this includes indicators such as arc light. In this case, we have a voltage-sensitive domain, or VSD, that is inside the neuronal membrane, and it's coupled to a bright fluorescence protein that is on the inside of the cell in the cytosol. And when there is a voltage increase in the membrane, there's a conformational change that ultimately leads to a change in fluorescence intensity. Other indicators, such as the ASAP indicators that have been developed by my colleague Michael Lin, have the same kind of 4 transmembrane domain, voltage-sensitive protein domain coupled to a fluorescent protein on the outside of the cell. And in this case, the ASAP indicator undergoes a decrease in brightness with membrane depolarization. Now, in my lab, we've been pursuing another class of voltage indicators that was first introduced by Adam Cohen's lab. These are indicators that make use of rhodopsin family proteins. These are 7 transmembrane domain proteins. We've been coupling them to bright fluorophores, again, on the inside of the cell. And like ASAP, these indicators also undergo a decrease in fluorescence output upon membrane depolarization. The FRET-opsin indicators that we've been exploring, as I said, couple a fluorescent protein to a voltage-sensitive domain from the rhodopsin family. And these proteins, these membrane proteins, have the interesting property that they undergo a change in their light absorbance as the membrane depolarizes. There's a change in the opsin protonation state. Now, by themselves, the archirhodopsin proteins can function as voltage indicators. They have good signaling dynamic range, as you can see here, but they turn out to be very dim fluorophores. And so for this reason, we've been coupling these rhodopsin family proteins to bright genetically encoded fluorophores. And in my lab, this was work done by Yang Gong, who now has his own group at Duke University. And so unlike the archrhodopsin family proteins, which often require much higher illumination intensities, we've been performing optical voltage imaging with milliwatts worth of light. So the FRET stands for fluorescence resonance energy transfer. We are looking at the much brighter fluorescence that we get from the donor protein, in this case a bright genetically encoded fluorophore. And there is an interaction with the acceptor protein, the rhodopsin family protein, such that when the membrane voltage depolarizes, there's a decrease in the fluorescence we get from the donor as the acceptor protein is able to essentially bleed off more of the energy, and it does not get emitted in a radiative fashion. And there has to be good overlap between the spectra, the emission spectra of the donor and the absorbance spectra of the acceptor for this to work well. Here are recordings with an earlier voltage indicator based on an L. maculans family rhodopsin protein. You can see on the left the fluorescence response of the rhodopsin to voltage depolarization steps is dim fluorescence. When we couple it to the bright fluorescent protein M-citrin, we can see the negative going change in the FRET opsin indicator as the opsin diminishes the output of the donor protein. Our recent versions of these FRET opsin indicators now provide records of the neuronal membrane voltage dynamics with high fidelity. As you can see, it is an optical record of a cell's dynamics on the top, electrical recording on the bottom. This is taken in vitro using milliwatts worth of light. Here on the left, you can see a construct that we use in our most recent indicator, coupling a fluorescent protein, M-neon, one of the brightest fluorescent proteins available, to a rhodopsin family protein from the green algae, acetobiolum, which we abbreviate ACE, much easier to say. And these images on the right show you that we're getting membrane-localized changes in fluorescence upon depolarization using 15 milliwatts of illumination per square millimeter. And you can see here, in comparison to the spectra that I just showed you previously, that the ACE family, ACE rhodopsin protein, has a better spectral overlap with the emission spectra of the M-neon green than the prior indicator that I showed you. ACE also has millisecond scale voltage-dependent kinetics, as you can see here in the optical responses to step depolarizations. If you look here at the finer time scale, you can see that the fluorescence output decreases. These are inverted on the 1 millisecond scale. Again, using modest illumination levels, much less than you would use with opsins by themselves. And the signaling dynamic range is about 20% delta F over F per 100 millivolts of voltage change. And this provides reports on subthreshold membrane voltage dynamics as well as spiking dynamics. Here you can see some comparisons between optical traces and electrophysiological recordings, dual recordings from neurons in culture, in brain slice, and in live mouse. On the right, you can see some quantifications of the D prime values that we were able to obtain with these recent ACE indicators in culture. You can see that the ACE2N and neon green indicator has a D prime value that's actually over 100. And this is going to be important because our D prime values are going to diminish as we go into more challenging experimental situations such as live mice or live flies. And we need very high D prime values, as I was saying earlier, to be able to keep our false positive rate low as we sample the data at kilohertz frame rates. This is data taken from an awake mouse. Layer 2-3 neurons were labeled, and you can see we're able to detect individual spikes. You can see the temporal insets showing you the data at finer time scales. And these are visual cortical neurons that lie in visual cortical area V1, projecting to cortical area LM, homologous to V2 in the mouse. And you can see these neurons are direction selective, which we can ascertain in the awake mouse. Dual optical and electrophysiological recordings using loose patch electrical recording methods showed that we're able to detect essentially all action potentials, and we can do so with a spike timing precision of about 200 microseconds, so far better timing precision than we get with calcium imaging. This is a movie showing our ability to reconstruct subcellular and submillisecond voltage propagation dynamics within a neuron in a mouse. This is trial average as opposed to the other data that I was showing you, which was single trial. We have similar capabilities for probing neuronal dynamics in the intact fly brain. This is data from Chung Huang in my lab. You can see the results here of a dual recording, optical and electrophysiological. And again, as in the mouse, we can detect neuronal spiking as well as subthreshold facets of the membrane voltage depolarization using modest illumination levels. Here's a close-up. And again, we have 200 microsecond spike timing precision. When we present odors to the fly, if we're performing optical voltage recordings from olfactory neurons, we can detect spike rates up to about 75 hertz or so and characterize the olfactory neurons' responses to odors and their odor tuning curves. And again, we can see intracellular propagation of voltage signals. This is a case where we were able to record from the dendrite of the neurons as well as the axon, and to see the slight 4 millisecond delay between the two structures in the same cell. So we're not quite at the point of being able to do this in freely behaving animals, as I showed you at the beginning with calcium imaging, but we are doing it in awake mice and awake flies, and we are hopeful that with continued progress, we will be able to perform the kind of studies I showed you with calcium, but using the novel optical voltage indicators. So to sum up, I discussed how we're using signal detection theory to guide our development of new indicators and the importance of trying to find indicators with a low background. Our modest improvements in the optical domain can lead to exponential improvements in spike detection error rates. We've been exploring the Fred Hobson indicators with their millisecond scale kinetics, D prime values over 100 in vitro. The ACE2N-amnion indicator now allows detection of single action potentials in awake mice. Timing accuracy is about 200 microseconds. We're looking at different facets of dendritic and subcellular voltage dynamics, as I just showed you, and we're hopeful that ahead of us, we may be able to attain cellular level imaging in freely moving mice using these new voltage indicators. And with that, if we have time, I'll be happy to take questions.
All right, thanks a lot, Mark. So I'm going to turn it over to Chris and Andreas. We have a couple of minutes for questions.
Okay, Mark, this is Chris. So I just found it interesting you said that with the voltage indicator with a D prime number, you can actually nicely calculate sort of where you can do scanned image and non-scanned image. You see, I have a slide on there. I think that's very interesting. Right? You said that within shallow range, you can do scan image on non-scan wide field image. They're actually very much the same.
Well, it's not a universal truth. We have done, and I should say that's also with calcium imaging. We have done some comparisons.
Yeah.
So, for example, in some cases, we have done imaging experiments with the same cell type and compared the D prime values that we have attained with one photon fluorescence imaging versus that we get with two photon fluorescence imaging. In other cases, we've even looked at the very same cells, not just the same cell type, and we've found very comparable D prime values. So it's an illustrative lesson that even though one photon imaging generally has far reduced contrast compared to two photon imaging due to the lack of optical sectioning, that nevertheless we can attain comparable D prime values, and this is because we're not scanning. We can sample each pixel for the entire frame duration rather than a small fraction of the frame. So we're working with many more photons.
Yeah. I think that's very interesting because that basically says there's certain depth basically you want to go to scanned version, right? As you go shallow enough, you actually non-scanned. You can live with the background generation basically.
That's right. Up to a limited depth, we can do very well and take advantage of the parallel acquisition across pixels.
Yeah. Andreas, you want to give a question? I can give that.
That was great, Mark. Thanks a lot. I have a question. It was very impressive, the voltage, and extremely exciting, especially for those of us that love measuring voltages. So I have two questions. One is, do you see a path that you'll be able to measure absolute, the level of the membrane potential with these indicators? And the other one, what do you think is the time frame, for development to go to population imaging using these methods, maybe by targeting specific compartments in different cells, so one can, let's say, go to what you showed at the beginning with the calcium imaging of hundreds of neurons, but measuring voltage and spikes instead, very fast.
Right. Thanks so much, Andreas. So the first question concerns absolute voltage measurements. Can we attain absolute numbers on the neuronal membrane voltage using this optical approach? So in the domain of calcium imaging, we learned that tracking absolute values of the intracellular calcium concentration is quite hard. There are approaches that have been successfully used in well-controlled experimental situations, particularly in vitro. There are strategies that involve ratiometric measurements, for example, or FRET indicators, or lifetime measurements. And it is conceivable that similar approaches might work in the future with the voltage indicators. Again, looking at parameters which are more robust and resilient to noise sources. However, there is going to be a substantial challenge from noise factors that occur in awake behaving animals, such as brain motion, hemodynamic effects, and so on and so forth. So even in the case of calcium imaging, where the temporal requirements are more modest, it is very challenging to get absolute numbers, and it has been done successfully in vitro. So I think what you're asking is going to be possible perhaps in theory, but is going to be very challenging in practice to pull off experimentally. Now let's see, your second question
was about dense imaging.
I'm sorry?
It was about dense imaging, where you label many cells, dense imaging with the voltage sensitive stuff you presented. What do you think is the time frame for that? And also on the imaging side, the technologies that you would recommend?
Thanks for clarifying. The web broke up for a second. So at the moment, there's some limitations. They come in some different forms. So, I stressed in the talk the importance of keeping the background photon flux relatively low. That was very critical for our ability to detect single action potentials in the awake mice. So what we did was we used a virus which enters axons. We injected this virus into cortical area LM, and then we imaged in cortical area V1. And so by injecting the virus non-locally, we were able to keep the background photonic flux low, and that was crucial to maintaining our D prime values. So that's one consideration, and it's one that the density of cell labels is one that may limit our ability to sample optical voltage signals from dense populations of cells. Another potential limitation is the fact that the voltage indicator decorates the entire cell membrane, at least as how we're presently using it. And in fact, even during action potentials, a substantial portion of the optical voltage signal that we're acquiring is in fact coming from the dendrites. And so when we look at the brain slices, if you have dense label, it's very hard to discriminate the processes of individual cells given a highly dense voltage label. So as I think mentioned, it may be beneficial to target the voltage indicator to different subcellular compartments, such as the axonal hillock, such as the soma, or dendrites in the cases where we're interested in dendritic signaling. And this may be an important part of future voltage imaging experiments. Now, another area of challenge is actually in the optical hardware. So to attain the kilohertz records of optical spiking in our paper, in our recent science paper, we had to restrict the field of view. And even if we had been able to maintain the current labeling density, there was not a scientific camera that I'm aware of that would have allowed us to sample the entire field of view that was afforded under the camera chip at that kilohertz rate. So part of the limitation we have is actually in the hardware domain. We need scientific grade CMOS cameras with broader field of view that can still attain kilohertz frame acquisition rates for broad populations, even if we make no strides forward at increasing the labeling density. So some of the improvements, to sum up, come in different domains. The last one is in the optical hardware domain. Some of it is in the cell biological domain, targeting indicators to appropriate subcellular compartments. And the third one is in the labeling strategy, helping to keep the background photon flux low. So those are the areas that we need to continue working on.
That was excellent. Thanks a lot. Thanks, Mark.
Thank you very much, Mark. I'm going to have to step in, and I want to thank Chris and Mark, our two presenters, and I also want to thank Andreas, our discussant, for two really good presentations and some great discussion this afternoon. Just to let everyone know, our next webinar will be held on Friday, June 17th in 2 weeks, and you can look for more information about that coming next week. And with that, again, thank you for logging on, and have a great weekend, everyone.
Okay, thank you very much.
Thanks, Matthew.
Thank you. Bye, everybody.
Thanks. Bye-bye.
Bye, Andres.